Maria Landinez, welcome. So Maria comes to us from tRNA Bio, and we'll be speaking about development of therapies for tRNA synthetase related CMT2D. Thank you.
Thank you so much. So I'm very grateful to the organizers for giving us the opportunity to present our work here. This has been a collaborative effort between Externa Bio and the Donders Institute. And what we are focusing on is to develop gene therapies based on tRNA. And today I'm going to present the work that we are doing on CMT2D. So as a very short introduction, you all know this way better than I do. Charcot-Marie-Tooth disease is caused by degeneration of motor and sensory axons and is genetically heterogeneous. There are more than 100 genes that have been causally linked to CMT, and from those, the group of the tRNA synthetases is the biggest family of genes that have been causally linked to the disease. From them, in our lab, we work with GARS, with the GARS gene, which encodes for the glycyl tRNA synthetase. And mutations in the glycyl tRNA synthetase are the cause of the CMT2D subtype. So what are these tRNA synthetases? These are the enzymes that catalyze the first step in protein synthesis. They bind tRNA to the respective amino acid, and later they release the charged tRNA for protein synthesis. What the lab has done in the last couple of years is to elucidate the molecular mechanisms of mutations in the glycyl tRNA synthetase. And this has been collaborative work with the lab of Rob Burgess. What we have learned is that in the wild-type situation, we have an enzyme that can bind tRNA, glycine, and release the aminoacylated tRNA. But in presence of CMT-associated mutations in the GLRS enzyme, there is a change of kinetics in this enzyme. The enzyme can still bind tRNA, it can bind the glycine, but it fails to release it. The enzyme is sequestering this tRNA, and the overall effect of that is that while in the wild-type situation, we have a big pool of charged tRNA that can be used for protein synthesis, then in the presence of the mutations in GLRS, this pool is reduced. And it's reduced so drastically that we find ribosome stalling specifically on glycine codons. And this activates the integrated stress response. So what we propose is that the root cause of the disease is the tRNA sequestration by the mutant enzyme. And we rationalize that to circumvent this tRNA sequestration, we can overexpress tRNA. Our idea is that if in the cell there is now so much tRNA available, even though part of that is sequestered by the mutant enzyme, still the wild-type isoform can release enough charged tRNA. You should remember that these patients are heterozygous and they have still a wild-type allele that can release charged tRNA. So we hope that the amount of charged tRNA in the cell will be going back to the baseline levels, which will allow for normal protein synthesis. And to test this hypothesis, the lab developed mouse and Drosophila models that were done by transgenic animals to overexpress tRNA. We first did that by inserting several copies of tRNA genes in the mouse genome, and that led to a twofold increase in tRNA expression, in glycyl tRNA expression. And what you can see here is that in the GARC201R model of CMT, these animals have a very strong motor performance defect that is fully rescued by tRNA-GLY overexpression. You can see that the animals overexpressing tRNA-GLY in the dark pink lines, they go back to the wild-type levels, and these animals are basically indistinguishable from wild-type animals. So at this point, we had a proof of concept that the increase of tRNA-GLY could work as a therapeutic strategy for CMT2D, and this was the starting point for Externa Bio. Then the goal of the company is to translate this to a therapy that can be used. So The lab also showed in Drosophila and in other mouse models of mutations in GLRS that tRNA-GLY overexpression could prevent all the CMT-associated symptoms that we measure in these animals. And what is also interesting to mention is that preliminary data from the lab suggests that mutations in other tRNA synthetases could have a very similar molecular mechanism, meaning that tRNA overexpression could be a therapeutic strategy for several subtypes of CMT. And then to bring this to a therapy, Externa Bio is developing AAV9s to deliver tRNA glycines. We chose the AAV9 serotype because it has been used in associated motor neuron disease, and it has been shown to be able to transduce motor and sensory neurons. We were using a control construct that is driving only expression of GFP. And then as a therapeutic construct, we have tRNA-GLY GCC with an ubiquitous promoter. Then we first wanted to test the efficiency of that in newborn animals before symptom onset. So we did injections at P0 via intracerebral ventricular delivery. And we measured the motor performance, electromyography, and histology at the end of the study. What we found is that with this dose and route of delivery, we are transducing around 80% of the motor neurons, and we are reaching about 1.5-fold increase in tRNA-GLY expression. And this increase was enough to produce a therapeutic effect. What you can see here is that the GARS animals treated with the control vector, they show these motor performance defects, which is fully prevented by tRNA-GLY overexpression. And furthermore, we did EMG measurements to assess the neuromuscular transmission. And here you can see that also tRNA-GLY overexpression can fully prevent the decrease in the compound muscle action potential and also in the latency of the response. So this was all very exciting for us, and that led us to move further in the development. And what we wanted to address next is what will be the effect of this therapy after symptom onset. I didn't show the data here for time reasons, but we also did histology to assess the axon degeneration phenotypes, the muscle denervation phenotypes, and the response of the integrated stress response. And all of that is fully rescued by tRNA-GLY overexpression. So in the next step to test if this therapy is efficient after symptom onset, we tried again with AAV9 capsids. And there, unfortunately, although we could reach high levels of motor neuron transduction, we could not reach high levels of tRNA-GLY overexpression. And what we need to show is if a tRNA-GLY overexpression can have a therapeutic effect. So we decided to use a different capsid that has been shown to transduce motor and sensory neurons with higher efficiency than AAV9. For that, we chose the PHPV and the PHPS capsids. They can cross the brain-blood barrier in black 6 mice. And we were using the same constructs, GFP as a control, and tRNA-GLY with an ubiquitous promoter. We are using again the GARC201R mouse model, which has a motor performance symptom onset around P21. And we are doing the injections at P30, 1 week after the symptom onset that we can measure. And we follow up these animals for 4 months after injection. And what we found was that indeed administration of AAV tRNA-GLY can revert the symptoms in these animals. What you can see here is that the animals at 4 weeks of age, they already have a strong motor performance defect. And we did the injections of the viral vectors right after this first motor performance point. And 2 weeks after injection, at week 8 of age, at week 6 of age, the animals are starting to recover. And by the time point of 12 weeks of age, they are going very close to the wild type levels. So we, besides this motor performance test, we also did electromyography, where we observe that the overexpression of tRNAGly can partially rescue the decrease in the compound muscle action potential, and it can fully rescue the latency of the response. So this altogether shows that tRNAGly overexpression has the capacity of reverting CMT-associated symptoms in these mouse models. And I want to show you the journey that we have in tRNAGly. So everything started in 2021 when we provide proof that tRNAGly sequestration is the molecular mechanism for CMT-associated mutations in the GlyRS tRNA synthetase. Later in 2023, we could show that administration of a vtRNAGly before symptom onset can fully prevent the onset of CMT symptoms. And by help of NLC Health Ventures, the Donders Institute, and the Radboud University, we could create tRNAGly, a spin-off from the lab that is focusing on further developing this therapy. In 2025, we could show that administration after symptom onset can revert the CMT-associated symptoms in our mouse model. And what we hope to do by the end of the year is to test the efficiency in a different mouse model that has an even more severe phenotype. And we also want to determine what would be the latest time point that we can do application of the therapy and still have a therapeutic effect. And as I mentioned before, we are now using these PHP-V and PHP-S capsids that only work in black 6 mice. So the goal for the next year is to find a capsid that can work for primates. And for that, we are open to collaborations, and I will be very happy if I can talk to some of you later to see if you have some more ideas on which capsids we could use. And that would be the main target for next year. And after that, we can continue the development. So we are a very small company. We are just starting. Right now, we are 4 people in the company. Next week, we will have a new scientist starting with us. And of course, this work would have not been possible without the help of all the lab, all the Storkebaum lab, also the lab of Jos Verhagen in Amsterdam, who is helping us with the production of the viral vectors, and our collaborators, Rob Borges and Scott Harper.